Neurons have what's known as an all or nothing response. If the stimulus meets the threshold level, the neuron will respond completely. If the stimulus does not meet the threshold level, nothing will occur. The response is all or nothing. So the impulse is actually going to be conducted if the stimulus meets that threshold intensity or is above it. All of the action potentials will be carried with the same strength. And the greater the intensity of the stimulation, the more impulses per second you will have. It is not that the neuron will have a stronger action potential. You will have more impulses per second, not a stronger impulse. After the neuron has been stimulated, there is a very short period of time where a threshold stimulus cannot produce another action potential. The resting membrane potential, or MP, has to be restored before the neuron can be depolarized again. So it's like a little tiny mini break. It's an absolute refractory period where nothing can happen to the neuron because the resting membrane potential has to be restored before it can be stimulated and depolarized again. You need to know about conduction velocity. The speed of an action potential is going to depend on a couple of factors. One is the axon diameter. The larger the diameter of the axon, the faster it will conduct an action potential, because the larger the diameter, the less resistance there will be. The amount of myelination is also going to play a key factor because myelin acts as an insulator and it allows almost all of the electrical charges of an action potential to stay within the axon. So it really speeds the rate of conduction up to 250 or 300 miles an hour. We're going to classify axons in 3 groups, and they will be classified on the diameter of the axon, how much myelination is there, and the propagation speed. The type A fibers are the largest of the axons in terms of diameter, and they will also be heavily myelinated. They can be up to 20 microns in diameter, which doesn't sound like much, but for a neuron, that's a lot. They will be motor neurons. They're going to control skeletal muscles, which means that's why we have such rapid responses when you want to move your skeletal muscles and contract them. Will also include somatosensory fibers, which supply your skin, your skeletal muscles, no surprise there, and joints. These guys are going to carry sensory information about position, balance, think proprioception, delicate touch, as well as pressure. And these are the guys that are going to carry info up to 300 miles an hour. The type B fibers are sort of like the mama bears. They are smaller. Diameter is between 2 and 4 microns. They are lightly myelinated, and they carry information at about 30 miles an hour. They're going to carry info about what's listed there: temperature, pain, and general touch, and pressure. They will carry information to smooth muscle, cardiac muscle, glands. Those are the effectors of the autonomic nervous system, by the way, the peripheral effectors. Type C fibers are the little guys. They are the smallest and they are unmyelinated, so their diameter is going to be less than 2 microns. They carry information at about 2 miles an hour. They will also carry temperature info, as well as information about pain, general touch, and pressure. These are the ones that are often going to carry information about chronic pain. They will be carrying information to peripheral effectors, which are the smooth muscles, the cardiac muscles, and glands. Remember why we want to have chronic pain information carried on fibers like C fibers, the type C fibers. If you carry chronic pain information on the type A fibers, you will be overwhelmed with pain and unable to function. Carry chronic pain information on smaller fibers so you get like a little drip at a time as opposed to a fire hose. What can impair conduction? The things that are listed there: alcohol, sedatives, anesthetics, because they're going to actually reduce the permeability of the membrane to sodium ions. So if you don't have sodium rushing into the cell, there will be no action potential. Cold, as well as continuous pressure, will start to affect whether or not you can have an action potential, because your circulation of blood is going to be interrupted. So you're not getting oxygen, you're not getting nutrients to the neuron, and therefore you are less likely to have an impulse or reach threshold in order to have an action potential. We need to talk about saltatory conduction, because saltatory conduction is very important, because saltatory conduction occurs in myelinated axons. When you have an unmyelinated axon, the action potential has to travel the length of the axon. That is slow. When you have a myelinated axon, and I'm going to update this PowerPoint so it doesn't say NI and nerve fiber, it's going to say AP for action potential and axonal fibers, you have what is known as saltatory conduction. It looks as if the action potential jumps from node of Ranvier to node of Ranvier to node of Ranvier. It doesn't jump. It actually travels under the myelin sheath. But what happens is you have very fast transmission of action potentials. So let's take a look at this. You are in the peripheral nervous system, and how do you know that? Because they mention Schwann cells there. So when this was first understood, when this concept was first understood, they couldn't figure out what was going on because they didn't have particularly sophisticated testing techniques. So it looked like the action potential was jumping over the myelin sheaths to node of Ranvier to the next node of Ranvier to the next node of Ranvier to the next node of Ranvier. What actually happens is, and write this down because you need to know it, what actually happens is this. There is a local electrical current that is carrying the action potential underneath the myelin sheath. So every time the action potential hits a myelin sheath, it's like that action potential has enough momentum to speed along underneath the myelin sheath until it gets to the node of Ranvier. At the node of Ranvier, the sodium and potassium gates will open and sodium will rush in and potassium will rush out, and then the sodium-potassium pump will reestablish the appropriate concentration gradient, building up more momentum in essence until that action potential hits the next myelin sheath. That local electrical current that is generated will carry that action potential underneath the next myelin sheath to the next node of Ranvier. All the activities associated with the real, in quotes, action potential will occur in the node of Ranvier. Enough momentum will be built up in terms of a local electrical current to carry the action potential under the next myelin sheath, etc., etc., etc. Fundamentally, what happens and what you need to know is myelin sheaths speed the rate of transmission of an action potential up to 300 miles an hour. And this shows you what's happening. Local electrical current is carrying that action potential under the myelin sheath. At the nodes of Ranvier, you have all the action potential stuff happening until you hit the next myelin sheath, and then you're going to have another local electrical current carrying information to the next node of Ranvier until you get to the distal end of the axon.